Hi, my name is Yunlong Liu. I'm the director for Center for Computational Biology and Bioinformatics. I will be teaching this module, Next Generation Sequencing 101. Here's the outline. I will give a very brief introduction about next generation sequencing technology and go through some major platforms and availability at IU Medical School. And I will review some of the very basic major applications. There are several keywords about next generation sequencing technology. So the first keyword is sequencing. It can sequence both DNA and RNA. Actually, most of the sequencing platforms sequence DNA only, but some of them will sequence the RNA molecule directly. The second keyword is short reads. So if you look at this DNA sequence molecule, it's not sequencing from the very beginning all the way to the end. Rather, it only sequences the very beginning part of it. Each element is called a read. It depends on different platforms. The length of the read can vary from very early on, 35 bases, sometimes even shorter, and all the way to today, about 300 base pairs. And sometimes we got a sequence, we can sequence both two ends of it. In this case, we call it either paired end or mate pairs, depends on the protocols to be used. I have to mention that some of the sequencers, especially the third generation sequencing, are not short read technology. Some of them can be very long, including PacBio and the nanopore technologies. The third keyword here is ultra high throughput. We are not sequencing this one molecule at a time. Rather, in one machine run, we can sequence up to 10 billion sequencing rates of those. So you can think of the throughput is super, super high. Now, a couple of leading platforms. The first one, I would say, has to be Illumina technology. This is an absolutely leading platform at the current stage, and it will include the bridge amplification and the sequencing by synthesis. So I'm not going to go through all the biochemistry part of it, but instead I'm going to give you some link and talk about what are the biochemistry behind these machines, why they are sequencing that fast. The second technology I labeled here as obsolete, and which is a solid technology. But I have to say that because it is being obsolete in a couple of years, for the past couple of years, but we still have some data that is in the public domain as well as in the publications. So you just know that there is a sequencer available as a solid, but it's no longer available. The third one here is the 454 technology by Roche. This is the reason I mentioned it here is this is the first ever commercially available next generation sequencing ever made. It's based on the parasequencing technology. It gives them longer rates, initially about 400 bases, eventually to 800 to 1,000 bases. And it is the first platform, but it is out-competed, so it's becoming obsolete. The next one is still hanging around, and it's very active, actually. It's a PacBio technology, or Pacific Biosciences. There are several very interesting features about this technology. The first one is it is a single molecule technology, which means that you get a molecule, you sequence it, you don't need to amplify those molecules, which have some of the advantage during the process. It is a nano-based technology, and the major advantage of this technology, it will create very, very long rates. Sometimes it can be up to 20,000 bases, or actually by average 20,000 bases, they can go much, much longer than that. Another one you read literatures there occur a lot, which is iron torrent iron proton technology. And actually we still have that machine in the Center for Medical Genomics here. They are semiconductor-based technology, and they do have a little bit higher rates, especially around the homopolymer regions. But this is a very interesting technology that sometimes that we serve as a orthologous validation platform for Illumina data. The last one is the nanopore technology, which is showing here. And if you look at this picture, it is in this person's hand. And that is actually the size of the sequencer. It is very portable. It is a single molecule technology. It's fast, cheap, and easy. A lot of people consider that is the future of the next generation sequencing technology. But regardless of this, Illumina is still the gold standard at the current stage that producing huge amount of data in the public domain. This is the whole family of Illumina sequencers. Of course, there are new ones that are started being made, but from very small fashion, mini-seq, and to MiSeq, NextSeq, HiSeq, HiSeq X, or NovaSeq technology. So the part I labeled as red dot are the ones that we actually have here at in the Center for Medical Genomics at IU Medical School. This is the whole fleet of our sequencing capacity. As you can see, this is quite a lot of sequencers. We have 2 NovaSeq 6000 platforms, and they have several different running modes, from the smallest chip that can give you about 800 million reads in one single instrument run, all the way to 10 billion reads. We have HiSeq 4000, NextSeq 500, MiSeq, and we also have live technology on proton sequencers, as well as the nanopore Minion platform access. So all these sequencers are available as a fee-for-service basis, and we are hoping that we will be able to working with all of you in the future to help with your experiment. In addition to the sequencing platforms, we also have single-cell technology, which is fueled by the 10x Chromium systems. And this is very interesting technology. We hope that we have a couple of modules that are specific talking about the data, the experiment, and all the way in between. But to be short, that this will give you a single cell resolution of transcriptome profiling that, as you see, it can sequence from 100 to 10,000 cells in one single assay. And it can do both single cell and the single nucleus RNA sequencing, and also single cell ATAC-seq, copy number variation, and single cell VDJ segmentation. As you can see, this is very important for immune system with the current COVID-19. I'm sure that a lot of people are very, very interested to use this technology and to figure out what's going on with the patient who can recover very quickly, and for the ones that eventually has to be hospitalized. This slide shows you the major applications that are enabled from what we have as a sequencing platforms. That was including the DNA sequencing, which is include the whole genome, whole exome sequencing, gene panel sequencing, and the linked arrays sequencing. RNA sequencing, we can do mRNA, total RNA, RNA from degraded FFP samples, and microRNA sequencing. To support the gene regulation, we can do ChIP-seq, CLIP-seq, ATAC-seq, and also for chromosome looping, 4C and high-C technology. For the DNA methylation, there are several different assays that we can do, including the METO-CAP-seq, as well as bisulfite-based sequencing. We can do single-cell analytics. We have been supporting a lot of metagenomic analysis, including 16S sequencing and shotgun sequencing. We have been also supporting a lot of technology development, and that will include various insertion site detection, CRISPR screening, and high throughput reporter assays. So this is the end of this module. Starting from next one, I'm going to start to talk about very basics on the next generation sequencing data analysis. We'll start from the data format. I hope you like it. I will see you next time.